prokaryotic cell division is going to be a little bit different from what we had in eukaryotic cells. In eukaryotic cells, we had mitosis and cytokinesis, but in prokaryotic cells, there is no nucleus. So since there is no nucleus, it's not going to be possible to have mitosis. However, we do still want to replicate the contents of the cell. And by replicating the contents, we're talking about the DNA and then all the other cellular components that would be inside. And we want to distribute those into two cells. We don't usually call these daughter cells that we're producing because for most prokaryotic organisms, they are unicellular. So these two cells that we produce, they're going to be just new organisms. So you go from having one cell to producing two new cells, which are two new organisms or two new individuals. This process here, as we're going to talk about it, this would be considered a type of asexual reproduction. And asexual reproduction is characterized by getting things that are exactly alike. So in this case, we are producing clones. So we're going to go through and we're going to talk about this asexual reproductive cell division that we have in prokaryotic cells. And this is going to be called binary fission. So what we'll find with binary fission is this is going to be simpler, and it's also going to be faster than what we had taking place in eukaryotic cells. So in a typical prokaryotic cell, there is going to be one circular chromosome. So I'm going to draw it right here. So since it's just one circular chromosome, that will simplify the overall process of division. We will only have to copy the one chromosome, and we won't have to put so much effort into distributing a lot of different copies of chromosomes into the two new cells that we're producing, the same way that we had to do with the eukaryotic cell. That circular chromosome, it does have some proteins associated with it, but there will not be any histones or nucleosomes as we talked about them before. So there are packaging proteins. And the packaging proteins, they will help with some organization in prokaryotic cells. Well, what happens here when we're talking about the cell division process is that this circular chromosome, it is attached to the plasma membrane at one location. And very near to that location where it is attached to the plasma membrane, there is what we call an origin of replication. And the origin of replication, well, just as the name kind of describes, this is where we're going to start copying the DNA. And in this case, there's only the one chromosome to copy. So if this origin of replication was right here, then at that one location, we're going to start to make a second copy of the DNA so that you'll actually end up with two rings of DNA inside this single prokaryotic cell. Now, as it's going to happen, this cell is going to elongate, and each of those rings of DNA is going to migrate towards an opposite side of the cell. So they do pull apart and separate from each other, and the origin is going to help with that, help them migrate in different directions. And then once the chromosomes get kind of far enough apart from each other, what we're going to see is that this overall large cell is going to begin to kind of pinch in half. So it will start to get pinched in the middle. We will still, again, have our chromosomes that are separated already on opposite sides. And eventually this is going to pinch completely apart so that we get our two new cells from this. So we'll have two cells here. And these two cells each will have one singular circular chromosome inside of them. And those circular chromosomes that they have are going to be identical to each other. So here's another quick look at this process of binary fission. Up here in the top, this would be the starting cell. And the first thing that the starting cell really needs to do is it needs to copy the DNA. So here we see the DNA getting copied. So we end up with 2 chromosomes in the same cell. So 2 of these circular chromosomes. And then notice in this picture, the chromosomes separate. And they do this as the cell continues to elongate. And then as the cell is pinching in two, and that's what we see right here, this is what we call a septum that is pinching and forming down the middle, and ultimately we end up with 2 identical cells.